As you start to explore the topic of reovirus, and this is the case for any scientific animation, you'll find that there's really an intimidating amount of information out there. And so I just wanted to point you to the primary literature. The images that I've shown you already, either the structure of this virus or the EM image, electron microscopy image, those sorts of images come from publications such as this one. So this is a paper written in 1993 by a team of scientists. And even though we won't go through the details of it, I just wanted to give you a feel for what this looks like. You may have seen one of these before. The title in this case is too long to read, but it has an abstract, and it's really a write-up of usually several years of research by a whole team of scientists. And so I wanted to show you this one in particular because it actually captures the theme of what our animation is going to be about. Specifically, they found that the virus in question, the reovirus, changes its shape as it's going through the process of infection. So whereas it starts as a relatively even sphere-like object, so it has many little protrusions, but it still looks like a sphere. As the virus progresses, as it infects our cells, it changes its shape. It undergoes a morph. And I'll come back to that later, but that's basically going to be the theme of our movie. And that is, how does the shape of the virus allow it to infect our cells? And what are some of these morphs that need to happen in order for the virus to infect us? Another publication, more recent this time, in 2000, this time using the technique that I mentioned before of X-ray crystallography, you'll see now that scientists are able to create much more detailed images of that very same virus. In this case, not only can they see the overall shape, but they have a sense of the position of every single protein in the virus, and even the position of every single atom within each one of those proteins. And you'll see some of these images get to be pretty detailed. So if you want to get the data that was used to create such an image, that's what's amazing, is that scientists in these fields all share the same resource to store and download this sort of molecular data. And that's called the Protein Data Bank. So the Protein Data Bank, or PDB, is a centralized database where every scientist deposits their data, and it's freely available to anybody. So you'll see at the moment there are over 70,000 known protein structures. I say protein, but actually this database includes other types of biological molecules, whether it's DNA, RNA, lipids, sugars. There are many other ones, even though proteins are by far the dominant ones. But the way you would use this is to simply enter your search term, in this case reovirus. So we're entering the name of a virus, but it could have been a protein. And the database will show you all of the different entries, in this case 24 structure hits that include the word reovirus. And you can see right away that these are, well, as you expected from the picture before, this is going to give you what's called a viral capsid, so the structure of a virus. So again, this database is for really any biomolecule. If you're interested specifically in viruses, I wanted to point you to another database called the ViperDB, or Viper database. And it lets you browse entries not only that are solved by X-ray crystallography, but also electron microscopy. It's a fun way to browse the great biodiversity of viruses in this case. So the last area before we move on to our next task is not just the shape of the virus, but also what is the environment relevant to the infection in our bodies. Reovirus, so the Reo of reovirus, REO, stands for respiratory enteric orphan virus, and it's that second word, enteric, which means the gut. So I just went and did a quick Google search for images related to the intestine or the gut. And what you'll see here is that there are structures known as villi, and these are protrusions of the lining of your intestine that actually increase the surface area and that give you a better ability to absorb food. So this villus here has a lining of little boxes that you can see in this diagram. Each one of these little tiny boxes is actually a cell. So if you zoom in, you'll notice that at the tip of those cells are yet more little protrusions, and these are called microvilli, and I'll show you images of those. It's the scale of the microvilli that are closest to the scale of our virus. So as we think of the environment to model behind or around our virus as it will start to infect our cells, this is the relevant structure that you want to be looking at, the microvilli. So let's just look at some electron microscopy images of villi. Pretty amazing structures. These are scanning electron microscopy, and then here, this is actually a cross-section. And again, you have to imagine that along these protrusions are many hundreds, probably thousands of cells, and each cell contains these other structures here called microvilli, these finger-like extensions that again extend the surface area through which food and nutrients can be absorbed. So it's a pretty amazing structure, and one that's kind of nice as a challenge for